Okay, so today I'll be talking about. So these are the person that I have covered. Last class we covered up to this one, and this is going to come in the exam, the last chapter for the midterm exam. And today I'll be dealing with these two, and this will come to the next final exam. So in last lecture we talked about the cell migration and neurogenesis, and how the cells are migrating to the cortex, how the radial glial cells helping the cells to migrate to the cortex, and how the neurogenesis happens. And today I'll be talking about one of the specific structure called as ganglionic eminence, and how this helps in migration of cell to the cortex. So before going to the specific things, so I want to so we want to know what is ganglionic eminence. So ganglionic eminence is a transitory structure in the development of a nervous system that guides cells, and also it guides axon to migrate. So here, so the structure looks like this, and here the cells basically they are migrating tangentially, perpendicular. So these are the red cells that you are seeing. These are the radial glial cells. So these green cells are the ganglionic eminence cells. So they are actually migrating to the cortical region, and they are tangentially they are migrating, and this is actually migrating perpendicular to the radial glial cells. And also some cells are also migrating parallel to the radial glial cells. So these are this structure helps in migration of cell in the tangential directions. So the ganglionic region, the eminence, is divided into three distinct structure. We have categorized into three distinct parts. One is the medial ganglionic eminence, which is called MGE, located here, and this is lateral ganglionic eminence, which is located here. And you have also the caudal, which is not shown. This is a coronal section of the mouse embryo, 12.5 days. So which is shown here. So the important thing that here you have to remember that the medial, in the next slide, if I'll show that this is again the sagittal section. So here the ganglionic eminence, so the cells are migrating from this region to the cortical region, and this type of migration is it's tangential migration of the cells to the cortical region. And in the ventricular zone, you have a radial migration of the cells to the cortex. So two distinct kind of migration. One is the tangential migration from the ganglionic region, ganglionic eminence region, and you have the radial migration from the ventricular zones that actually help that's how you have a formation of the cortex, because of the two distinct type of migration. So so the inhibitory neurons mostly, if I'll show that in the next slide, I'll come to the previous slide. So mostly the GABAergic neuron, the inhibitor neurons, are derived from the ganglionic eminence. Okay, so they show tangential migration. So mostly in the medial ganglionic eminence region, those inhibitory neurons, the GABAergic neurons, that migrate tangentially, perpendicular to the radial glial cells, and they migrate to the cortex. And the pyramidal neurons or astrocytes, they migrate radially to the cortex. So that's how you have formation of a cortex because of two distinct kind of migration. One is the tangential migration from ganglionic eminence, and another is the radial migration. And these pyramidal neurons, they migrate radially to the cortex, or in the case of GABAergic neuron, they migrate tangentially from the ganglionic eminence region to form the cortex. Okay. So some cells later also become oligodendrocytes that derive from the medial ganglionic eminence region, and some of the cells in the lateral ganglionic eminence region, they migrate radially to become neurons of the striatum. Okay, so here two different kind of migration has shown here in this picture. So so here you can see that these cells are they are moving. This is the radial glial cells, and the neurons are migrating in the process of the along the leading process of the radial glial cells. So this type of migration is called a radial migration, and this type of migration is called as a tangential migration. So this type of migration is seen in GABAergic neurons, and this type of migration that you are seeing seen in the pyramidal neurons. So the pyramidal neurons, they migrate radially, and the GABAergic neuron, they migrate tangentially to form the cortex. Yes.
In that image, it looks like the cells are tangentially, they join with the radially. Is that what happens?
Yes. They migrate. Here, if you see in this first picture, here in this picture, they are migrate tangentially, forming the cortex, and some of the cells also migrate like parallel to these radial glial cells. Yeah, they migrate in both directions.
Okay.
Yeah.
On Tuesday's lecture, you said the device to the beginner. Was it just throwing that?
Sorry?
Like on Tuesday or the last lecture, you said that the radial glia divides to the progenitor, and the progenitor goes up radially. So are you saying that the cells that are coming tangentially just joins that back?
It depends on which group of neuron you are talking about. Here is the GABAergic neuron. They show this kind of movement. Yeah.
So it's different.
Yeah, different neuron. Yeah. Okay. So, yeah, the same thing is kind of I talked about the GABAergic neuron that show like first tangentially they move, then also they move along with the radial glial cells vertically. Here. So this is all about the ganglionic eminence that I have covered. So these are some of the important points. I will recommend that you will go over the slides and remember those things. So here some of these points I have mentioned, like migration is guided by the radial glial cells, and mostly the ventricular zone produces the projection neuron. So I have written the like key points for the cortical development, and the glia are the last cell to be born. So some of the other key points that I have also mentioned, like radial glial cells are the progenitor cell for the neuron. So the study from has been shown that radial glial cells are the progenitor cells, and the migration is guided by the radial glial cells. So all these points key points I have mentioned. I will recommend that you go over the slides. At least remember some of these points so that you know this might come in the exam, in the future exam, in the final term. So we'll proceed for the last section of this neurogenesis. So I talked about neural crest, neural tube formation, and how from the neural tube the neural crest are generated. Then you have generation of different subsets of a neuron. So we'll go cover the last part of this session. So here, before going detail to the mouse model or other higher model system, I just want to emphasize little bit about neurogenesis. So before we talked about the embryonic neurogenesis at the during the embryonic development. So here I want to emphasize on adult neurogenesis. So there are evidence that neurogenesis happens in the adult stage. So we'll start with the basic model, the Xenophos, the frog, and we'll proceed for the mammals, the mouse. So here the two stages of frog development has been shown. This is the larval, this is the tadpole stage, and this is the matured stage, the frog. So the frog eye has been a great model system to understand basic mechanism of neurogenesis. So here in the tadpole stage, you can see that the eye is a little bit smaller, and in the adult frog, the eye is bigger, and that because of division of cells and specification of formation of diverse neurons and other cells. So here, if you look at this tadpole eye, so there are specific group of cell present in this eye that is called as stem cells, and they are also called as ciliary marginal zone cells or CMZ cells. So they have the capacity to produce different sets of neuron in the retina, and when the frog develops, the tadpole develops in the adult frog, you can see that the eye size is bigger, And though this happens because there are those stem cells which are present also get they are dividing in the adult stage, and that we called as adult neurogenesis, and that happens and happens in the adults. But there are several studies which says that this adult neurogenesis is not well kind of studied. This is going on. So the prior hypothesis is that neurogenesis happens during the embryonic stage, and the cell they are the terminally differentiated cells. They don't divide. But later people found that neurogenesis also there are specific reasons where neurogenesis happens, like in adults, in the human, there is a specific reason in the dentate gyrus, that is the hippocampus, where neurogenesis has been shown. So in adult, neurogenesis happens. You can see that in the tadpole eye also, the size the eye is getting bigger because you have the stem cell like cells are present in this eye, which is dividing to form different subsets of neuron. Okay, the retinal neuron. This is another example of adult neurogenesis in mouse. So here, this is the part of the brain has been is shown here. So you can see that this is the during the embryonic stage, the neurogenesis happens in the ventricular zone. So I talked about that, how the things happens in the last class, last lecture, from the ventricular zone. So you have something like that in the adult brain, that is the reminiscent of the ventricular zone, that is the lateral ventricles. So here the lateral ventricle, you have the specific type of cell, stem cell, which helps in neurogenesis, or which helps in the formation of a neuron in the adult stage. So this green type of cell that you are seeing here is the B cells, which I'll be talking about soon. So here, what happens is the cells are produced here in the ventricular or subventricular zone, and they migrate to the olfactory bulb. Okay, and this happens during the adult stage. And here, this is the mechanism how the cells formed and how they migrate. So what happens is, so this specific type of cell, the green cells that you are seeing here, this is called as B cells, and the B cells has a process that is projecting to the left lateral ventricle. So there is a ciliary-like process present in the B cell, which is projecting to the lateral ventricle, and also another process which is projecting to the blood vessels. So the B cells divides to form C cells. Okay, the larger cell, the red color cell that you are seeing, this is the C cells. The B cells divides from C cells, and C cell divides to form smaller cell called as A cells. Okay, the A cells actually migrate from this subventricular zone through the process called as rostral migratory stream to the olfactory zone. Okay, and later they differentiated into periglomerular cells and periglomerular cells and granule cells. So this thing happens during the adult stage, and we call this as adult neurogenesis. Okay, so in human we have the hippocampus and dental so there is the this is the reason where you have this neurogenesis happens. So here this is another example to show that the neurogenesis happens at the hippocampus. So here what it has been done that, so the GFP is cloned in the retrovirus, and this is injected to understand that the neurogenesis happening in the adults, and also the neurons are showing similar property like like the progenitor cells. So they have done this experiment where they have cloned the GFP in the retrovirus, and they injected that GFP to the mouse, and the mouse is given certain task, so that this mouse is kept in a chamber where it is continuously rolling, like so it is given certain task. Then we want to know that whether because of performance of a certain task, whether new neurons are getting generated or not, and these neurons showing similar property like progenitor cells. So the neurons, so as I mentioned, that hippocampus is the region where the neurogenesis happens, and this is the center for learning and memory. So because since the mouse learned certain tasks, So we want to see that whether the neurons are generated also. So it has been seen that the neurons are formed because these are GFP labeled, and those neurons showing similar physiological property like granule cells. Okay, so that means neurogenesis happens in the adults, and they show similar property like progenitor cells. Okay. Any questions? Okay. So this is all about the neurogenesis, ganglionic eminence. Then we'll move to the last part of this, neural tube and neural crest. So then how different cells are produced from the neural crest, different diverse kind of cells like neuron and other cells. Okay. So this is the part of the development of nervous system during the embryonic stage. So here, this is during the neural tube closure stage, and this is a pre-neural closure stage. So you can see that you have, the mass of cells that generated either side of the neural tube. This is called as pre-soma, and this is derived from the mesoderm of the embryo. This later become somites. Okay. So these are the mesodermally derived cells which are present either side of the neural tube, and also it plays an important role during the development of nervous system. Okay, so you have generation of a special group of cells called as neural crest cells from the in the neural tube, which migrates and forms different sets of neuron. And okay, so this is the neural tube closure stage. So you have a generation of foreplate, and which happens because you have formation of another mesodermal structure called as a notochord. Notochord gives signal to the neural tube. So you have initiation of floor plate, then you have initiation of a roof plate, and the generation of a neural crest. Okay. Now this structure, pre-somatic mesoderm, this pre-somatic, they become somite, and this is also mesodermally derived structure. Okay. So then you have generation of a roof plate. Okay. So next, what happens is the migration of cells and differentiation of a cell from the neural crest. And the migration of cells from the neural crest depends on signal, and this is specific type of signal called as afferent signal. Okay. So you have these are the somites. Okay, these are the different somites. So this is the anterior portion of the somite. This is the posterior portion of the somite. So you have anterior, posterior. Okay. So this is the signal afferent, which actually determine where the neural crest cell will migrate. So afferent basically potentiates anterior migration of a neural crest, and it suppresses the posterior migration of a neural crest. Okay, so that has been shown, and somites are the precursor population of cells and give rise to important structure like it forms skeletal muscle, it forms cartilage, it forms tendons, and also somites also determine the migratory path of neural crest cells and axons of the spinal nerve. So somites has so many rules. Okay, so this afferent signal actually regulates the anterior migration of the neural crest cells. Okay, so just kind of remember that afferent is the signal. Now this is just in the lab, it has been shown that how afferent regulate neural crest cells. So you can see that when you have afferent signal, it regulates the migration of neural crest cells. So you have the anterior migration of neural crest cells, but when the posterior half migration is inhibited. So this is the mechanism how the migration happens in the neural crest cells. First, what happens is, so here is the detailed steps for the neural crest migration. So you have epithelial cell close to the neural crest cells, and they undergoes epithelial mesenchymal transition phase. So you have generation of afferent signal, So here you can see that it migrates in the anterior part. Anterior part is the rostral part. Okay. So afferent signal, helping the migration of neural crest cells to the anterior, anterior part, that is the rostral part. It inhibits the migration of neural crest in the posterior, like the caudal part, the posterior part. Okay. So this is just schematically shown that how afferent regulate neural crest migration. Then neural crest migrate to different part of this during the development, and so you have cardiac neural crest, which forms the structure associated with the heart. Then you have enteric neural crest migration, so it forms structure associated with the gut. Then you have trunk neural crest. Then you have cephalic neural crest, cranial neural crest. So neural crest cells are multipotent stem cells. It forms different structure, including neuron, different structure of the body during the development. So this is just shown that what are the different structure that are formed from the neural crest. So you have the generation of eye, you have olfactory, generation of olfactory system, then you have a generation of sympathetic ganglia, then dorsal root ganglia from the neural crest cells. You have a generation of glia, trigeminal nerve, everything. Okay. So these are the derivatives. So these are multipotent stem cells which can generate neuron and other tissue also. Okay, so this is an example or experiment shown in mouse developing in the mouse embryo. Okay, so this is a 9 days old mouse embryo. So here you can see that the neural crest cells are tagged with the GFP tag, and in the real time during the development, you can see that how the cells are migrating from anterior portion rostral to caudal, and so this is mapped with the GFP. So here the same thing is shown here schematically. So you have anterior part of the brain, that is this is the anterior part, this is the midbrain, this is the hindbrain. So just to show that how neural crest cells migrate to the different parts of the brain. So here this is one example showing that how crest cell migrate outward in an orderly fashion to pharyngeal arcs or fronto. So here this is shown that how the neural crest cell migrate to the pharyngeal arcs. Okay, like different parts of the during the development. So these are some of these points. Some of these diverse group of cells showing that how this special type of neural crest that is called as cranial neural crest. So in the previous I talked about like a trunk neural crest or enteric neural crest. So you have also the cranial neural crest. So it forms the different types of cells. So these are mentioned here, like it forms the parasynthetic ganglia, it forms the Schwann cells, it forms all different diverse kind of cells. Okay. So like different parts of the different type of cell that I have shown, how the cranial neural crest forms this until the cranial, all these different diverse type of cells, like the melanocytes and the Schwann cells, all these types of cells. So here, what are the fates for the neural crest? So as I talked about, the cranial neural crest from the anterior portion of the nervous system, the posterior portion of the nervous system formed by the trunk neural crest. Okay, so here the cranial neural crest consists of prosencephalon, the forebrain, and the mesencephalon, the midbrain, and the rhombencephalon, the hindbrain, and the posterior portion, mostly the spinal cord portion consists of the trunk from the trunk neural crest. Okay, so starts from like the spinal cord, like cervical spinal cord, thoracic spinal cord, or lumbrosarcal spinal cord. So these are formed by the trunk neural crest, and everything is like showing here. Why this is not moving? Wow. yeah, there are some. So this is the fat map showing in the trunk neural crest. So the green color that is so these are all color coded. Like the yellow that you are seeing for is parasympathetic ganglia. Mostly it is formed from the cranial neural crest from this mesencephalon. Okay, so all these are color coded, showing that fat map. Like you have two distinct kind of neural crest, like cranial neural crest from the anterior portion of the brain, or trunk neural crest from mostly the spinal cord, the posterior portion of the nervous system in the developing embryo. And this is the fat map showing that which type of neural crest is which type of neuron is derived from the which type of neural crest. So just go over this, and you will come to kind of know. So we'll not put any specific. You don't have to mug up anything. We'll not put any specific question that tell me this type of neural crest is formed from which this type of tissue forms from which neural crest. We are not going to put that type of questions, because this is something is not analytical questions. Just you should have an idea that do have different diverse kind of neural crest which forms diverse type of neurons or diverse type of tissue. Okay, so mostly the neurons and glia they are produced from these structures, like sympathetic ganglia, sensory ganglia. So this is just to show that diversity of neural crest, how to form different types of cells. So we'll go to the trunk neural crest fat. So again, during the closure of the neural tube, we have generation of different structure, four plate. The notochord gives signal to form the four plate. Then you have migration of neural crest, and also your formation of roof plate. So these are the four sets of things that is formed from the trunk neural crest. These are mentioned here, like the melanocytes formed from the trunk neural crest, or neurons and glia of sensory ganglia formed from the trunk neural crest. So these are the FATs. Okay, so here just shown in the diagram, showing that what are the different cells, different group of neurons which form from the trunk neural crest. So you have formation of melanocyte, showing the same thing that I have shown in the previous slide in a more simplistic way, like dorsal root ganglion cells, sympathetic neuron, adrenal medulla. All these things are derived from the trunk neural crest. Okay, so but the formation of neural crest fat depends on the local signal. So here the very interesting experiment has been done from two different species, quail and chick. So here I just want to emphasize that the formation of cells depends on the local signal. So I'll just to highlight that two distinct portion of this neural crest cells derived from this distinct portion of the developing embryo in the embryo. So one is this is the part, this is the cranial neural crest, and this is the trunk neural crest. So with the cranial, you have three distinct parts. You have prosencephalon, forebrain, midbrain, and hindbrain, and you have specific type of nerve present in each part of this developing neural tube or developing brain. So this here I'm going to talk about some specific experiment they have done to identify that the local environment actually helping in developing specific type of neurons. Okay, so here what they have done is they have taken some cells from the mesencephalon, and they put it in the in the trunk neural crest region, in the cervical spinal cord region. They want to know whether it is for it is behaving with this fat, or it is also behaving with this fat. So they just want to know if the transplant change these different types of cell to another position, how they behave. Okay, so here they have done experiment with two distinct neuron, that is parasympathetic neuron, which are present in the cranial neural crest region, mostly in the mesencephalon region, and sympathetic nervous system basically present in the trunk neural crest region. Okay, so what they have done is, when they transplant some of the cells parasympathetic in this from this region to this region, and those cells actually become sympathetic. The parasympathetic fat becomes sympathetic because you have the local signals in this region, which is actually helping to differentiate to the sympathetic fat. So the neural crest cells, generation of specific type of cells from the neural crest depends on the local signal that I want to emphasize. Okay, so more experiment has been done in the chick and the quill. So here again the parasympathetic. This is different species, chicks and quill. So this is transplanted from quill to chick, chick to quill. Then this is the sympathetic. This is the parasympathetic region, and they want to know whether from the different species whether there is any kind of changes. So here again they have transplanted from this to this, from this to this. So but the cell became depending on the local signal. So if you transplant from parasympathetic to sympathetic region, it will become sympathetic because you have the local signal which is actually helping to form the sympathetic neuron. Or if you transplant from sympathetic to parasympathetic, it will become parasympathetic. Okay, so it depends on the local signal. So here the same experiment, like transplanted from here to here. These are the so you have distinct group of cell present in this embryo. So you have the cholinergic neuron basically in the parasympathetic region. You have the sympathetic region. So when you transplant from sympathetic region to parasympathetic region, so these cells become cholinergic because you have the local signal that actually transforming cell to cholinergic, even in different species. Okay. So yeah, so what are the factor? What are the signal actually helping, transforming cells to different cell fate? What are the local signals? So we know that these neural crest cells are multipotent stem cells. So can anybody give me a specific answer, like what is the difference between neural crest cells and neural stem cells? What is the main difference between? Any idea?
Maybe the neural crest can become everything, but they can become a select type versus neural stem cells. They can become all of the nervous system if you give them the.
Exactly. So neural crest cell can form different diverse kind of cells. It can form the part of the heart. It can form the gut. It can form the neurons. But yeah, you are right. So they are multipotent, the neural crest cells. So so these are the multipotent stem cells. So you have different signals which actually transforming the multipotent, transforming the cells to different group of cells. So you have the BMP24, which helps in the transforming cell to neuron. You have the GGF, the glial growth factor, which form the Schwann cell, and you have the TGF beta, which form the muscles. So these are the local signal which is actually transforming the cells to different cell fate. So more closely, so if this is a very complicated thing, and a lot of signals are there. So these are the neural crest progenitor cells. So if you have FGF2, they become sympathetic progenitor. Then with NGF, they become androgenic neurons. So they become, with this factor, they become melanocyte, or glucocorticoids, they become chromaffin cells progenitor, which become chromaffin cells. So we have different kind of local signals which transforming the neural crest cell to different group of cells. Okay, so these are the signals. And some neural crest cells are predetermined. These are called as predetermined neural crest cells. So here the predetermined neural crest cells. The previously I talked about the multipotent neural crest cells, which become different kinds of cells. So that they are predetermined. So they also migrate, to form, specific group of specific cells, not group of cells, specific cells. So these are called as predetermined neural crest cells. So, so in the last section of my lecture, I'll be talking about what are the key transcription factor in the neural that is determining or forming different group of cells. So during the different stage of the neural tube development, you have different kind of a transcription factor which expresses and forms different group of cells. So this is the early stage of the neural tube closure. So you have this is called as neural plate border module. So this is the early stage of the neural tube development. So you have, these are the transcription factor which expresses in this stage, which is the PAX37 or GBX2. Then when the neural tube invaginates and kind of come to the mid stage of the closure, these are the transcription factor which expresses like FOXD3. You have SOX-E, PAX37 also expresses. Now the final stage is the neural tube closure stage. You have generation of neural tube, you have formation of notochord, and also you have migration of neural crest from the neural tube. Then you have expression of this transcription factor, and also some of the transcription factor they overlap. Okay, so this is all about neural crest. Is a very short lecture because most of the part I covered last lecture. So if you have any questions, I'm happy to kind of take all those things. Yeah.
So you said today's lecture is not on exam.
Yes, today's lecture is not on the exam. So so you have let me go to the, so the exam is from the previous lecture. I go to, so the exam will come up to this, from my like some part of the Ryan's lecture and some part of my lecture, like 6 lecture of mine, not 6, 5. So up to this. So 40 questions, 1 hour. I will suggest that come 15 minutes before so that we can have alternate arrangement, sitting arrangement. And don't take stress. It will come directly from the slides and very simple. So yeah, all the best for the exam. So no, it's not, homework is not multiple choice questions. Some of the questions I put multiple choice, and the descriptive type kind of question I put in the homework, but I have not put the questions which is coming from the homework. So this is different kind of question that I put, but they are all multiple choice and very simple. Just go over the slide, don't mug up anything. Very technical questions. I hope you guys able to do good. Any questions? Okay. So all the best for exam on Thursday, and prepare well. I hope you guys will do good.